I know what you're doing.
No. Okay, so let's learn how to do these dihybrid crosses here. So we're going back to Mendel. He's already established the principle of dominance and segregation. So he saw that some alleles are dominant over other alleles. He saw that the alleles segregate. So even if you have a heterozygous individual, you can still get the dominant or the recessive allele. So now he's going to be testing to see whether certain alleles always segregate together. So for example, if you have blue eyes, does blue eyes always go with blonde hair? Is what he's looking for. Obviously, we know the answer to that now. But this is what he's testing here. So he's looking in his pea plants here. If this pea is round, will it always also be yellow, for example? Or can we get mixes? So he's going to take purebred green wrinkled peas and cross them with purebred round yellow peas. So this is called a dihybrid cross. Remind me what the prefix di means? Two. And it's a hybrid, so a combination of the two. I will say that round and yellow are the dominant traits. So what do you think Mendel saw in his F1 generation? Round and yellow, because they are the dominant traits. And you can see this just with if we did individual Punnett squares. So if we do this here, we do purebred round. What's the only? So remind me again what purebred means.
It's like.
Isn't it like homozygous dominant?
Homozygous dominant. So that means all your offspring are going to get that same trait. But what else can you be? You can also be homozygous recessive. So actually what he has here, he's crossing plants that are homozygous dominant for both traits with plants that are homozygous recessive for both traits. So you can see here, 100% of these plants are going to be yellow, and 100% of these plants are going to be round. So this was no surprise to him. This is kind of what he expected. He knew it wasn't going to be until his F2 generation that he could see, can I potentially get round with green, or can I potentially get wrinkled with yellow, is what he's looking for. Okay. So then he's going to self-pollinate, or he's going to allow the F1 plants to self-pollinate. And he's going to look, will I only get round yellow and wrinkled green, or will I get combinations of the two? Of those F2 plants, he got 556 seeds. 315 were round and yellow. So you remember, round and yellow was a pea generation plant. So the grandparents of these. He also had 32 that were green and wrinkled like the other pea generation plant. He also got 105 that were a combination, round and green, and 104 that were yellow and wrinkled, another combination here. So what this tells him is that genes do not necessarily come together in pairs or come together in trios. Is it possible to have blue eyes, blonde hair? Yes. Is it possible to have brown eyes, brown hair? Yeah. Can you have blue eyes, brown hair? Yep. Can you have brown eyes, blonde hair?
Yeah.
Yes. So there's no set rule that separates those traits. And Mendel actually got very, very lucky with this because there actually are some traits that you can inherit that you also inherit with other traits. Usually those are going to be ones that are actually on the same chromosome because you inherit chromosomes together. So for example, let's see, color blindness is an X-linked trait. Is there another X-linked trait somebody can think of? Can't think of one off the top of my head now. But say there's two traits that are X-linked traits, one of them being color blindness. If you inherit color blindness, you're probably also going to inherit the other trait because they're on the same chromosome. The only way you would not inherit them together is if they crossed over right in between those chromosomes. And you'll actually, if you take college biology, you'll figure out how to calculate whether they are linked, and then you can actually calculate how closely they're together, how close they are on a chromosome. Good luck, that's not fun. A lot of statistics. But what Mendel discovers for these, since they are independent of one another, is this principle of independent assortment. So these traits are independent of one another, and they can assort in all sorts of different combinations. Okay. So because of this, we know that we can use mega Punnett squares like we talked about in our warm-ups. These 4x4 Punnett squares. So these are going to be 16 squares. We'll show you how to, we'll do one together here in a little bit. But in order to get all of your individual alleles, we have to use a specific technique. So you guys think, you can be, if you are heterozygous for everything, how many different combinations can you have there? Well, you can calculate. What's my chances of being capital R? 1/2. What's my chances of being lowercase r? 1/2. What's my chances of being capital Y? 1/2. So if I'm just looking individually, I have a 50% chance, whichever one I get, I have a 50% chance of one I get. That's actually a shorthand way, you just would have to flip it. There's 4 different combinations of alleles you can have here. How many of you, I think it's in algebra, you are talking about this right now? So how many of you are in an algebra class right now? Are using the FOIL method? How many of you have heard of the FOIL method? Okay. So, those of you that have heard of it, what does this F stand for?
First, outer, inner, last.
First, outer, inside, first, outside, inside, last. Yep. So, here are my combination. Here is a plant that is round, heterozygous for being round, and heterozygous for being yellow. What alleles am I going to have for that first, for the F here?
Capital R, capital Y.
Capital R, capital Y. It's the first letters in each group here. What about for my outside? Capital R, lowercase y, the letters that are on the outside. What about for inside?
Lowercase r, capital Y.
R, capital Y.
Lowercase r, capital Y, the two letters on the inside. And then finally for last,
R and
lowercase r, lowercase y, the last letters of each pair here. So this gives me my 4 different combinations here that I will need in order to correctly do a Punnett square, a dihybrid Punnett square. Okay. So here is what this would be set up as. So this would be parent 1. This would be parent 2. So the same as just our normal Punnett square. But each pair of letters now gets its own column. Do not, I say do not even though I know some of you are still going to do this and it's going to annoy the heck out of me. Do not do this. That is wrong. I see that all the time. That is wrong. Because what's going to happen here, do you do it over on this side too? I guess I should set it up this way. This will be a little bit better. This is actually more common what happens. You set it up this way. You get this. Okay, cool. I know that this P is round, but I have completely lost knowing what color it is. You have to have both letters here. So do not do that. Okay, but once you have your Punnett squares set up here, you still follow the same rules. This RY is going to go down here, and there, and there, and here. This RY is going to come across to each one. And do the same thing with each column and with each row. So how many letters should you get in each box? 4. You should get 4 letters in each box. Okay? So what am I going to put right here? Capital R, capital Y, capital R, capital Y. Now let's put this in a more traditional format. Capital, capital, capital R, capital R, capital Y, capital Y. If you want to do it the first way, you can, but when we start trying to figure out our phenotypes, it tends to get very difficult to read those. So what you can do is what I just did there, go RY, RY, and then go back and fix it into the more standard format. That's going to make it easier to find what you're looking for. Okay, what about this one here? Capital R, capital R, capital Y, lowercase y. What about here? And here? Lowercase y. Okay, let's go down here. should we go through the rest of it? Or we
No,
can
I
get,
think we got through it.
think you got it? Yes.
I got the rest of it.
Okay, so again, you follow your rules, you should end up with 4, and then from here, you can then calculate your phenotype ratios. We won't do the phenotype ratios on this here, but remind me again what phenotype is?
Physical
That
trait.
the physical.
The physical trait. So what is the phenotype of this one here?
Rounded out.
And
Round,
Yellow.
Yellow.
yellow. Let's go ahead, let's do all these across and down like we just did.
Round, yellow. It's round yellow all the way.
It is round yellow all the way across. Why is that?
because.
Cause rounded yellow is
Round
ours.
yellow.
Capital R, white.
Yep, because we have both dominant traits going across there. Guess what's gonna happen here?
Same thing.
Same
The
thing.
exact same thing, so round and yellow down through all
So
of it.
what's lowercase?
Lowercase.
That'd be green.
Square.
It'd be wrinkled and green.
Okay.
Wrinkled and green, yep. So you get those down
How
here.
do you know which ones to put first? So like, what if you do W and G?
So usually, so round is dominant, so
Oh,
we take the R from there. Yellow is dominant.
so take the yellow, or take the dominant trait and use it.
Take the dominant trait and use that letter, or take the like trait. So, say we wanted to do hair color, and we have blonde and brown. Well, those are both B's, so you could do a B, but some people might get confused on, trying to remember which one might be dominant in that case. So what you could do, or maybe you also have a B in another trait, you could also do H's for hair color, for example. As long as you choose one, and have it make a little bit of sense somehow, it doesn't take too much thinking to figure out how you got that letter. Usually, you can keep everything straight. Okay, we ready to do one on our own here? Okay, so what I want you to do, you're gonna do it on your own, and then check your Punnett square with a partner. And then you're going to come up with the phenotypic ratios that we see here. So, we're using Mendel's P-plants here. Tall is dominant to short. Smooth is dominant to constricted. And we are crossing individuals that are heterozygous for both traits. So, ignore the genotypes. Just give me the phenotypes and the correct Punnett square. Give you some time to do that. Once you have the Punnett square filled out, check with the partner, come up with your phenotypic ratios.
So we just put separate, there's two. So for, like, it's 75%.
16 altogether.
So it's 6 apples. So there's, for that. So there's heterozygous type. Because like, they can be a short court.
Yeah, but he's a
So about 39 is kind of so bad. I think taller than here though. Tallest dominant.
Is Centennial every week?
Like after
I
bed?
don't think so.
Or just don't?
I know, is it like Folgers don't?
Can move them.
I'll have to look at the roster here.
Cause I know Franklin doesn't have a big guy, neither does Centennial.
Holdridge will have one open probably, unless they lied on their roster.
And I'm like,
Where are you at? This one right here, is this one?
Okay, do I have a volunteer that wants to come fill out this Punnett square
I'll do it after I'm done here.
after you're done here? Okay, make sure that you're very careful counting your S's as well. Don't get your S's mixed
Oh,
up.
okay.
So like your little assistant.
doesn't look like that.
I need that to go up there, so.
No.
More like,
Mike, it's like
Make sure you're doing it right? No.
Should made a space skinnier.
Hey, come on.
Nice try, big boy. Make me spill when I kill you. Kid. Oh.
Yeah. Well, yes, but like.
Touch it, you die. Touch it, you live. Alright, Gavin, I'm done. You can have it.
Yeah. I just want one thing.
Is Hannah the one that has like lots around here? And works in
Yeah,
Somebody
she's
can help
getting
him out with
married
the
for that
phenotypic ratios as well.
75 and 5 years old.
What's this?
Sunny?
She quarantined?
I don't know.
Oh, I did not do this right now.
Definitely didn't realize I was in front of that, but.
How'd you guys do
I can't believe it was too much too quickly.
How'd you guys do it? I'm not sure.
Oh, that's that.
I mean, I got these. Yeah, I got those right. I just don't know, I'm not sure.
Do fractions.
So it was, I was like, 12 over 16.
It goes 9/16, 3/16, 3/16, and 1/16.
They did those just never really fun. Yes. Oh, it's a joke.
Alright, let's head back to our seats and figure out how much we trust Seth here.
A lot.
Super hot.
Yeah, you guess.
I trust Seth more. What?
Oh, wait. Oh, I know. Never mind.
Look good to you? Yes, no, maybe so.
Yes, it looks amazing to me.
Yes, so here's a trick for you to tell whether you're did this correctly. When we have two heterozygous individuals in a dihybrid cross, this only works for that. Because if one of them is homozygous for anything, this ratio does not pop up. But you will always have 9 dominant to 3 dominant recessive to 3 recessive dominant to 1 recessive. And is that what we got for our phenotypic ratios?
Mm-hmm.
Yes, so we can tell that we did this Punnett square correctly, because we got that same 9 to 3 to 3 to 1 ratio.
Are you gonna make it, or about the genotype?
Usually we don't do genotypes on this, but you could figure it out. It'd be a lot of 1/16, 2/16. You would get a 4/16 with your heterozygous ones through the middle there. Any questions on this?